Hello, my name is Tarek Hamdan, and for my project, I compared the amino acids at the receptor binding sites of the SARS-CoV-2 virus found in humans to 5 other organisms. You may be asking yourself, what is SARS-CoV-2? SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2, is the virus that infects an individual causing the disease COVID-19. The SARS-CoV-2 virus infects an individual through the fusion of the virus with the host cell. This is done through the binding of the spike protein found on the virus with the ACE2 receptor found on cells within the respiratory tract of humans. The SARS-CoV-2 virus attacks the lungs of an infected individual. The spike protein on the SARS-CoV-2 virus consists of 2 subunits, the S1 and S2 spike proteins, each with their own function. Simply put, the function of the S1 spike protein is to bind to the ACE2 receptor found in the host cell, while the function of the S2 spike protein is to facilitate the fusion of the 2 cells. For my experiment, I used the NCBI database to access the amino acid sequences of the SARS-CoV-2 virus for humans, as well as 5 other organisms, which include rhinoceros, Asian palm civet, African civet, tiger, and humans. After collecting the amino acids for each non-human organism at the receptor binding sites, I compared them with those found in the human SARS-CoV-2 virus. With this information, I was able to calculate the percentage similarity between the organisms and compare them. Given that the SARS-CoV-2 virus was able to occupy all 5 of my selected organisms, it was expected that they would all show high similarity to one another in amino acid sequences. Also, since the virus is believed to have come from a wet environment, it was also expected that there would be high similarity between the virus found in humans with organisms from relatively wet environments. After collecting data on the location as well as the types of amino acids present in the 6 different viruses, I created a table to visualize where the differences lie. Among the 5 organisms that were compared to humans, the SARS-CoV-2 virus found in tigers was the most similar to that found in humans, with 100% similarity. To no surprise, the virus from another organism within the same genus, the lion, also displayed high similarity to humans with 94.12% similarity. Both of the civets, African and Asian palm, showed 76.47% similarity. The rhinoceros showed the least similarities with only 70.59% similarity. The SARS-CoV-2 virus in humans is very similar to that found in other organisms because they work the same way. No matter the organism, the SARS-CoV-2 virus uses its spike protein to bind and fuse with the host cell. Because of this, only a few changes need to be made in the amino acid sequence of the virus in order to be successful in targeting the host cell of a new organism. There are various ways a virus can travel from one organism to another. Luckily, in the context of COVID-19, which is a respiratory disease, there are far less ways one could become infected than with other diseases. This, however, does not mean it is hard to become infected. The easiest way the virus can travel is through conversation and interaction in close proximity. The best thing we all can do to avoid infection is practice social distancing and wear masks when it is possible. Thank you for listening to my presentation on the SARS-CoV-2 virus. I hope you have a wonderful rest of your day.